Welcome everyone to week number 3 in Science 200, our Natural Sciences Exploration course. This week you're going to learn all about using scientific sources. Last week I told you use the Shapiro Library. It is a wonderful source of information for you. Google Scholar and PubMed, those are two databases that you can also search. You will not find a lot of full text articles in those, but in the Shapiro Library you will. So we're talking about scientific sources this week. All right, last week you got an understanding, a better understanding of how science actually works and the importance of data through the work of John Snow and his examination of outbreaks of cholera in London, England, and how he discovered that cholera was transmitted through contaminated water instead of miasma. So I hope that you enjoyed that story because it was really interesting, and it showed you how science works and the importance of discoveries and how discoveries are made through tedious work and through data examination. You can come to totally different understandings of the world because of things like that. And so that was exemplified by Dr. John Snow. This week is all about using scientific sources. The first thing that you have to learn is how to read a scientific paper. I will tell you, when you do your searches now, do not just do a Google search. Of course, all of us Google search all the time. I know I do. I guarantee that I do it every day. But it's looking for a restaurant or finding fun things to do this weekend or something like that. It's not looking for scientific sources. So I've already given you 3 databases to look at. Now I want to talk to you about how to read a scientific paper and the importance of the different sections. The Flint, Michigan water supply issue with all the lead in the water is the example that you're going to read about this week. It was a horrendous example of what goes wrong when people don't pay attention to what they're doing and try to cut corners and that sort of thing, and really have destroyed the lives of so many people because of it. And Flint, Michigan still has a problem with their water supply. So anyway, read all about that this week. I believe that I'm putting a video in the announcements on that too. All right, what I want to do now is try my best to open an example of a scientific journal article that I have found. So give me just a second to see if I can do it, because this is going to be interesting. Here it is. All right, here it is. I'm going to put my face over here. All right. So here is an article that I found. I hope you can see my face. I think you can. About using a particular gene editing technology called CRISPR-Cas9. It is a type of manipulation of DNA where you can go in at any point, any place in the DNA and make changes. This is a wonderful new discovery that has just come about in the last few years. Before that, there were only certain places in the genome where you can make cuts and where you can make changes. And this is much more targeted. You can go in at exactly the right place where you want to go and manipulate DNA. So this is a wonderful way to edit genes. What they're doing in this study is making virus-like particles, and that it will be the VLPs that you'll see later on, that can be used as a vaccine against COVID. So here is a new and better way, a more specific, more targeted way to educate your immune system about the SARS-CoV-2 virus. V stands for virus anyway, but this virus that causes COVID and helps protect our health against invasion by this particular virus. So this is just an example article, okay? We don't need to know too much else about the subject. What I want you to do next is look over here on the left. Here's an outline. Now, not every article is going to have an outline like this, but they all have the same sections, okay? I think it's just really good with this article that the outline is on the left so that I can go through and show you the different sections. All right, the most important section of the whole article. This article is about some research that's been done and it's been published in a scientific journal. The most important section is the abstract. The abstract is a summary of everything that this article says. Everything from introducing the idea to giving you the background information to telling you how they did the experiments to what the results were and to what that means. It's all right there in the abstract. I cannot tell you how important an abstract is. You can usually get enough information out of the abstract without having to go to the other sections. So please do look at the abstract in every one of these journal articles that you take a look at, okay? So if you think about it, I mentioned some of the sections that are in the article. There will be one or two sentences that sum up each one of these sections that I'm going to cover next. So next we've got the introduction. So what is the introduction? It's introducing the idea of this research, what it's going to be about, why it's important, and what everybody else knows about this particular topic. Now, if you will look down, look at all of the references as we go through this thing, right? So what they're doing, they've done a background search, and they've gathered up information from all these other sources and put them together to help explain SARS-CoV-2 and how their vaccines are made up until this point, and what we know about this virus. And then what the question is that these particular researchers are examining. Method and materials. Here I am over on the left again. Methods and materials. Uh-oh, where'd it go? There it is. I call this materials and methods. But anyway, the materials are never listed. Never, ever, ever. This is an explanation of what actually was carried out. And it tells you everything, like specific information about the mice strains. Look here. Mouse adaptation strain C57MA14. Even the type of mice that they used are in here. Exactly how much antibiotic is made and put into the cell culture liquids that they use to feed the cells in culture. So all the information is here about how they did the experiments. Now, why is this important? Because other scientists who are interested in this work are going to take a look at their methods and say, okay, what they have decided to do will answer this particular question, or, that's just what I'm working on. Looks very interesting. How have they tweaked it? How have they done this a little bit different? Have they looked at anything else that I haven't even thought about looking at? That's the materials and methods section. ELISpot, it's like ELISA, it's an enzyme assay. That's just one of the methods here. Then the results section, ta-da, the results, what did they get? So they're showing you here some pictures of cell cultures that they did, and then they lit up different parts of the cells and the virus particles that they had infected the cells with, and that's why you see all these different colors. Here are all the results of the experiments, and they provide, and you would, pictures, graphs, to show you what they discovered. You just don't say, oh, this is what I found. No, you put the data out there. And so lots and lots of data, including pictures, all right? And then we have the discussion section. The discussion section tells you what everything means, right? It's the author's interpretation of those results that they obtained. What do these results mean? And also in the discussion section, you will talk about the problems that you had, that you encountered, how you corrected for them, or produce controls out of them, or whatever you had to do. They'll also talk in the discussion section about new questions that have arisen because of this research and ideas about how to move forward with this particular topic. So that's your discussion section. Here are some other interesting things. I like this part here, author's contribution. So this tells you what each of the authors did in the particular study. Funding. Ooh, that's important. I'm going to mention that again in a minute in this video. Funding. So who paid for this? This is terribly important for scientific research. Who paid for it? Because is that going to cloud the results that you got? We have to make sure of this, and you have to be open and above board in telling us where you got your money in case there are any conflicts of interest. Authors, declaration of competing interest. Do you have any stock in a vaccine company and you're the one of the authors of this study? Oops, that might be a problem. Where is it? Declaration of competing interest. All authors declare no competing interest, right? Wouldn't that be what you want to see? And then, of course, the references. And references are very important. If you find one really good article that is helping you a lot, then take a look at their references. And you may be able to find two more instead of going through all that trouble of doing a whole gigantic search of the Shapiro library. So that's another hint. Okay, so that's where we are. All right, let me get back out of here. Whoop, and get back to my slide. So that's how to read a scientific journal article. It's actually pretty easy. What was the most important thing? Read the abstract. Okay, this week you are going to find 3 excellent scientific sources to help you answer your research question that you came up with in week 1. Okay? Now, what's the trick to this? You got to find credible sources. And that's one reason that you have to look at that funding issue, because that has been a problem through the years. Whoever pays for research usually gets the results that they want, right? Money speaks or money talks, however you want to say that, even in science. So the credibility of the source. If they're funded by the NIH, National Institutes of Health, then it will be excellent research, I'll guarantee you, because nobody is influencing the scientists to come up with a particular result. Relevant. Is this what you're looking for? Is this answering your question? If you're looking at ocean acidification and the article that you have gotten is talking about how trees are dying in the Amazon, okay, no, they're really not related. So a relevant source too has to answer the questions that you're looking for, credible and relevant. And like I said, you always look at that funding, always look at the end of an article to see who funded the research. You're going to find 3 good sources for your topic, and then you're going to evaluate them. And you're doing this mainly, I don't know why I'm way down here now. You're mainly doing this through your Sumo web text this week. All right. Plagiarism issues. Please be careful of this. I want to mention this. Don't use someone else's work in any way. You have to turn in your work through Turnitin, and I am telling you, Turnitin can catch everything. And if you have a high score in Turnitin, you can see the score just as well as I can. Okay? If you have a high score, the first thing I do is open your assignment and look to see where the similarities are. Now, sometimes it's the list of references. And since you have to use APA style and you use the same reference as somebody else, it's going to be similar. So sometimes it's just as simple as that. Sometimes the whole paper's been copied. So please be careful of this. Turnitin catches these. That's why you have to turn in your work through Turnitin. So just be careful of that. You have discussion board postings in week 3, and this is week 3. So what was the biggest challenge for you in analyzing a scientific source? And man, if you don't say right off the top of your head that the jargon in science is a totally different language, that's one of the issues, isn't it? You look at these scientific articles and it looks like a bunch of mumbo jumbo to you because the language is not something that you're familiar with. So you have to look those words up and see what they mean. You don't have to understand every method, but you do have to understand important words. So of course that's going to be one of your challenges. How did you overcome this? Use examples. Okay, like in that article that I just showed you, VLP. I didn't know what that was. I know all about viruses. I know all about genetics. I didn't know what a VLP was. Virus-like particle. If you can learn one thing about geneticists, they love some acronyms. Oh my word, they do. They'll abbreviate anything. They're like, sorry, that's my dryer. They're like people in the business world. They love acronyms. So anyway, I had to look up what VLP was. Virus-like particle? Couldn't you just say that? So anyway, so use particular examples. Use at least 2. And make sure you use the correct format for any of your discussion board postings. Read those instructions. I can't emphasize that enough to you, those guidelines, right? Your 2 response posts, you're going to talk to your peers about the challenges that they said that they faced, and you're going to say whether you had those same challenges or you had different ones from them. And share tips for analyzing any type of source or doing any of these searches. Share tips with us. We can all learn from each other doing this. Okay? You don't have anything else to do with your research project except find these 3 sources, okay? And like I said, you're going to do that through the textbook. You're going to enter those into the textbook in Chapter 3, and I will give you some feedback on those through the textbook. Tip of the week, use what you learned in the textbook to answer the discussion board questions, because there's a lot of good information there about scientific sources. And follow the directions for completing your assignments, and that means look at those guidelines. The guidelines are the instructions. The rubrics are the grading sheets, how it will get graded. And they go hand in hand if you look at them. The guidelines are found in the rubrics. So look at the guidelines and rubrics. All right. I hope that you have a very good week this week. There's my email, m.sigmon@snhu.edu. Please let me know if you need anything. I'm here for you. I'll answer your email questions as quickly as I can. Have a great week.